Good evening, folks. I'd like to welcome you to our virtual fall SRC meeting and rules update for the 2021 State Science and Engineering Fair. I'm going to address just the basic rules to get us started, and then each of the other fire team members will deliver the information on each of the sensitive areas. We're going to start out with a discussion of science ethics, since we're fortunate enough to have several members on the SSEF, SRC, and DNS. We've been notified that SSEF is going to focus on ethics this year, and I'm really glad. It's an area that is tough to deal with, but that we have had problems with in the past, because it covers things that you might not consider strictly ethical. And there's an outline here from the SSEF rules on the areas of project research that they consider falling into this particular area. The point they make is that without good ethics, there is no good science, basically. And our goal here is to keep our students safe, to keep those around our students and our environment safe, and to encourage the production of good science research. And the only way to do that is to do it ethically. Each year we have issues involving questionable dates, mentorship, the actual participation of the students. What did they do on their own compared to what did they claim to have done by putting it into a procedure or as part of the research plan? Did they follow the rules of science fair, both SSEF and state rules? Did they pay attention to ethical treatment of their human subjects, or good husbandry of their animals, or appropriate skills and aseptic technique when dealing with PHBAs? There are many different aspects of science research that apply to ethics. From time to time, we find that the ethical violations may not be just the student. It may be uninformed adults who don't understand the rules, and many times don't understand that there's a difference between pre-collegiate research and the research of college or university students. So we need to emphasize the fact that students who are undergoing science research under the mentorship of professionals need to make sure that those mentors have access to the copies of the rules, so they understand the rules that they have to abide by. Every now and then we find a date that is curious, maybe something that looks like it's been erased and changed on an approval form, or something that's not clear. And we will ask the students to produce a logbook. More discussion on logbooks later, but that is one way that all scientists validate their research, is the use of a credible logbook. Unfortunately, sometimes the practices that fail to follow the ethics code of SSEF, sometimes we have to disqualify those students. It's unfortunate, but it does happen. So we want to try to nip that in the bud and pay a lot of attention to this up front. and realize that the students have to sign an ethics code, basically, with SSEF. So they need to be aware of the importance of the practice of science ethics in good research. Project eligibility. Project eligibility is one thing in this era of COVID that has already come under question and will continue to. There's nothing fair about the fact that these students did this research last year, were prepared to go to their regions or as a state finalist or as an SSEF finalist, and then we're not able to do that. But we have no control over that. So we have to abide by the rules that are established for eligibility. That means that the only way that the students can use the information from a project that was performed for last year's fair is if the current project meets continuation policies. So if it is truly a continuation this year and not just a replication of the project, then they will be able to use last year's data, last year's processes. But the research project has to be significantly different this year than it was last year. It cannot just be a repeat of last year's project. It has to meet continuation requirements. Knowledge of these requirements is the responsibility of the students and the adult sponsors. So when we're counseling our students on what they should and shouldn't or can and can't do, eligibility needs to be a big part of it. Because of the scrambling of educational situations this year, we've already had several questions about who can participate. The students may participate if they are part of a Florida educational program. Doesn't have to be public school, doesn't have to be face-to-face. It can be homeschool or it can be virtual education, but they have to go through a selection process by competing in their local fair and be selected to participate at the state level. So as far as eligibility goes for participation, those are the rules that we have to follow. I have had a question about the participation of a student in a team project who moved out of state. And so that student would not be able to participate in this year's research. They have to be a Florida educational student. Affiliated fairs such as the state fair or your regional fairs must follow SSEF rules, but they may establish rules that are more strict, as we have done here in the state of Florida, and all participants under that umbrella, the competition of the state science and engineering fair, must follow our standards and guidelines. So it's important that they have those available to them. Those are available on our website, our state science fair website. So make sure that students who are participating have access to those rules. Forms. The first thing when we come to forms is make sure that you have the current forms, the correct forms. They change from year to year, and some forms may not have the current information required, and therefore the student would not have filled out the correct information and their forms would be incomplete. The year of participation is written at the bottom. It's typed at the bottom of the form. So make sure you have the forms that go along with the 2020-2021 Science Fair season. The first 6 bullets on this list cover forms that are required of every project, not facsimiles, not handwritten substitutes. You have the official SSEF entry form, the official abstract, the checklist for adult sponsors, student checklist, the research plan, which includes the bibliography, and Form 1B, which is most of the approval forms for SRC. When you look at the information that's required on these forms, the students have to be aware that they need to fill them out completely and legibly. These forms can be word processed, or the print-on forms can be accessed. Every now and then we get handwritten forms. If they're not legible, we are not going to be able to approve the project. So make sure that the forms that come out of your regions to state are legible. The adult sponsor can be a teacher or a parent or someone who is aware of science process. The students need to be aware that all the questions on these forms need to be filled out. We're going to talk about the research plan a little bit more in just a minute. Other forms as required by the specialty of the project. If a project is done at a university or a research lab somewhere that is an RRI, the Form 1C needs to be filled out. One of the important aspects of filling out these forms is to make sure that the right person signs them on the right dates. They don't all predate research. This is one of the ones that has to be filled out after the student research is done. The qualified scientist, Form 2, or the designated supervisor have to be people who have knowledge of the practices that the student is exercising during their research. The risk assessment needs to be filled out with awareness of the risk to the student, the other people around them, including the subjects, the animals that are involved, if animals are involved, and the environment. Human participation must be pre-approved through an institutional review board, an IRB. The constitution of that IRB is critical and very well laid out in SSEF rules. We've run across in the past problems with the membership of the IRB where they don't meet the qualifications of SSEF rules. Unfortunately, it's the student that pays for this, even though it's not their problem. The informed consent will be discussed at great length by Joe Scott, who's going to make the presentation on humans. Vertebrate animals. There are 2 forms that relate to vertebrate animals. Vertebrate animals done in the field or at home or at a registered research institution would be, in the field or at home or at school would be the 5 A's, and the ones that are taking place at an RRI would be the 5 B's. You must include, with any vertebrate animal form, you must include the state's mortality report that will keep up with the subjects that died during the research process. They must undergo a necropsy if they die during that period of time under the student's stewardship. And a vet must do the necropsy and decide whether the animal perished as a result of the research or not. And that decision must be made before the research can proceed. If it's decided that the death of the subject is due to the research, then the research must come to a halt and will not qualify for competition. The PHBA questions will be covered by Will, and he'll cover the differences between BSL-1 organisms and BSL-1 procedures, and the same with BSL-2. Risk assessments must deal with the possibility of contamination. Aseptic technique and disposal must be addressed. Human and animal tissues, human and animal tissues come under the general heading of potentially hazardous biological agents, but are dealt with on Form 6B. If the student receives the tissues only and doesn't participate in harvesting the tissues, then they don't have to include other forms. But if the source of the tissue is a vertebrate animal and the student is involved in the collection of those tissues, then they have to fill out the vertebrate animal forms. The continuation forms are filled out for those projects that are multi-year research projects. And again, they have to indicate that the direction of research has taken a fork, a detour, and is not the exact same project done just with a different set of variables or with different instruments. Dates and signatures. I've mentioned that the Form 1C, for instance, is not one of the forms that has to be filled out prior to research beginning, but that it is a form that reviews the research and the student's involvement in the research and must be filled out by the mentor at the RRI. Other dates must precede the wet work or the actual hands-on research that the students are doing. Dates have to fall within the acceptable parameters of research. Research, which is literature search, may begin before the actual date of the wet work, but the approval for the project has to be in place and dated before the hands-on or wet work begins. Mentors are a choice of the student researcher and must meet the requirements of individual projects. Adult sponsors can be anyone who is going to be assisting the student with understanding the rules or the procedures or their day-to-day work with the information for the project. Adult sponsors can be parents, teachers, or any other adult. Qualified scientists must hold an advanced degree in the area that is being researched. It has to be in the appropriate area. If a student is doing behavioral research and has human subjects involved, then the qualified scientist must be degreed and educated in the area of human research. If the student is working with vertebrate animals or PHBAs, the advanced degree must be in those areas. Many times teachers will get a master's of science education. While it's laudable that they get an advanced degree in education, that does not qualify them to be a qualified scientist in the area of a pure science. The designated supervisor is the person who oversees and has knowledge of the procedures that the student's carrying out. If the qualified scientist is overseeing the day-to-day research, then the student doesn't have to have a designated supervisor. But a qualified scientist is only required if the reviewing board of the SRC or the IRB determines that they're necessary. If a student is doing research at several different venues or in different stages, they can have more than one qualified scientist and more than one designated supervisor. The research plan. This is probably the one area that we have the hardest time getting over to the students, the appropriateness of the form of the research plan. There is a template for a research plan questionnaire with ISEF's paperwork. The thing that we try to stress with the student is that you should be able to hand your procedure, your research procedure, to any other person who has the same skills that they do and have them be able to replicate their research. It's got to be extremely detailed. You have to use the correct measurement units. You have to use the correct nomenclature. And you need to include only those things that you directly performed, not what a lab group did or what a major professor or primary researcher did, but only those things which you participated in. If you as a research student are using instruments in a lab, then the procedures for using those instruments have to be detailed and outlined within the research plan. The only alternative to writing out those steps of the use of the PCR equipment or even a microscope or an incubator or a SPEC 20, in lieu of writing out the procedures that you used for each of those, you can reference in the bibliography a methods manual from the lab that you used. But one of those two things has to be covered, either the procedures or the reference within the research plan to those procedures. And please have them not submit their research paper as a research plan. It's much more appropriate to have a stepwise sequential procedure to follow so that you can replicate the steps in research. The bibliography is a requirement. Make sure they have the correct number of bibliographic references. And in the case of the sensitive projects, they must have methods citations from each of those different areas. Logbooks. Good logbooks, and I stress good logbooks, appropriate logbooks, have been responsible for getting students out of many holes that they've dug for themselves in erroneously reporting when or what they've done. A good logbook is a diary detailed about what you do as a researcher every day. And it's dated and arranged sequentially and chronologically usually in a bound notebook. Now, in this day and age of technology, we are going to accept logbooks that are digital as long as they can be date and time stamped. Logbooks are extremely important. And as I've already said, if we run into a problem with a date or a procedure or a contact, the logbook is one way the student can show us what actually happened. Projects exempt from review. SSEF has a list of exemptions in several different sensitive areas. We have chosen at the state level not to recognize any exemptions in areas of vertebrate research, human research, PHBAs, tissues. Any of these sensitive areas we would like to review or have reviewed by a local SRC or IRB prior to research. So if you're using zebrafish, you still need to get the zebrafish projects reviewed. If you're doing behavioral projects with either humans or animals, please have them reviewed. If you're doing PHBA, which cannot be done at home, have them reviewed. I understand that there are documents out there that say that E. coli K12 is not a pathogen. That is not an overall understood and accepted idea. If you're working with bacteria or other microbes, get the project pre-reviewed before you start your research. Afterwards, it's too late. All projects carried out at a registered research institution must have approval of that research institution's board. In order for that institution to be used, in order for its facilities to be used, it doesn't matter if the student has a parent who is in charge of the lab, they still have to be approved. The signatures and dates affixed to those documents of approval may be transmitted digitally, but they have to have a timestamp, and they've got to have the information in them that is required by SSEF or Florida State Science and Engineering Fair. We get projects every year that sort of make us scratch our heads. And most of you regional directors have been on the SRC before, and you've reviewed these projects, and you notice that they are woefully incomplete. We don't understand how they could have gotten through at least 2 levels of review and come to us in the state that they come. And it makes our jobs, it makes your job a whole lot more complicated. Please stress with your local science fair coordinator, please stress with your regional committee that review these papers that they've got to read them as if they were on the state SRC. Is the state going to approve this project? If the answer is no, or I don't know, that's a good time to call one of us that are on the SRC. It's a good time to go back and review the rules and ask yourselves, is this a project that I want to put a signature on as approving? If not, turn it around and deal with the student or the teacher or the parents. At that level, it will be a whole lot easier. They will be a whole lot less aggravated than if you wait and pass it on to state and we have to tell them, we're sorry, this project does not qualify. We, the members of the fire team, and you have our email addresses on the state supplement, we are completely open to assisting you before the review time or anytime during the year as a run-up to the state science fair. It will make all of our jobs a whole lot easier if we solve problems while they can still be solved ethically. We have a huge job, and the job is made much easier if we jump on things ahead of time. And hopefully we will have success in doing that. Thanks to your participation, thanks to your volunteerism, hopefully we'll have another successful year. Thank you.
Hello. The following rules apply to research using hazardous chemicals, devices, and activities. These include substances and devices that are regulated by local, state, country, or international law, most often with restrictions of their use by minors such as DEA-controlled substances, prescription drugs, alcohol, tobacco, firearms, and explosives. Hazardous activities are those that involve a level of risk above and beyond that encountered in the student's everyday life. Documentation of approval and safety precautions are key elements when working with hazardous chemicals and devices. These rules are intended to protect the student researcher by ensuring proper supervision and the consideration of all potential risks so that the appropriate safety precautions are taken. Students are required to meet all standards imposed by ISEF, SSEF, school, local, and other regional fairs. Please note that SSEF and RSEF rules and standards supersede ISEF rules and standards. Forms 1, 1A, and 1B are required of all projects. Hazardous chemicals and devices projects will often require one or all three of Forms 1C, Form 2, and Form 3. Form 1C, Regulated Research Institutional Industrial Setting. When work is conducted in a regulated research institution, industrial setting, or any work site other than home, school, or field, during the current ISEF project year, the Regulated Research Institutional Industrial Setting Form 1C must be completed after experimentation. Form 2, Qualified Scientist. A qualified scientist is required for all studies involving DEA-controlled substances. Qualifications must be earned a doctoral professional degree in a scientific discipline related to the student's area of research, and or individual with experience, extensive experience, and expertise in the student's area of research must be thoroughly familiar with the regulations that govern the student's area of research, may live elsewhere and not be local to the student, in which case a designated supervisor has been appointed and trained to serve as the on-site supervision as necessary for the specific student project. Form 3, Risk Assessment. The student researcher must conduct a risk assessment in collaboration with a designated supervisor or qualified scientist prior to experimentation. The risk assessment should be documented in the research plan to include the risk assessment process, supervision, safety precautions, and appropriate methods of disposal. The risk assessment is also documented on Risk Assessment Form 3. Notice that this assessment must be included in the research plan and on Form 3. A risk assessment documented on Form 3 must include proper disposal methods for chemicals used in an experiment. A risk assessment Form 3 is recommended for all student design inventions or prototypes. Additional rules for specific regulated substances. DEA-controlled substances. All studies using DEA-controlled substances must be supervised by a qualified scientist who is licensed by the DEA for use of that controlled substance. All studies using DEA Schedule 1 substances must have the research protocol approval by the DEA before research begins. Schedule 2, 3, and 4 substances do not require protocol approved by the DEA. Prescription drugs. Prescription drugs are regulated by federal or country laws to protect against inappropriate or unsafe use. Students are prohibited from administering prescription drugs to human participants. A veterinarian must supervise student administration of any prescription drugs to vertebrate animals. Alcohol and tobacco. The U.S. Alcohol and Tobacco Tax and Trade Bureau, TTB, regulates the production of alcohol and distribution of alcohol and tobacco products. Fermentation studies in which minute quantities of ethyl alcohol are produced are permitted. The designated supervisor is responsible for the acquisition, usage, and appropriate disposal of the alcohol or tobacco used in the study. Production of wine or beer by adults is allowable in the home and must meet TTB home production regulations. Students can design and conduct a research project under direct parental supervision involving the legal production of the wine or beer. Students are prohibited from conducting experiments where consumable ethyl alcohol is produced by distillation. Students can distill alcohol for fuel or other non-consumable products. The work must be conducted at school or an RRI and follow all local and country laws. Firearms and explosives. Projects involving firearms and explosives are allowable when conducted with the direct supervision of a designated supervisor and when in compliance with all federal, state, and local laws. A fully assembled rocket motor, reload kit, or propellant modules containing more than 62.5 grams of propellant are subject to the permitting storage and other requirements of federal explosive laws and regulations. Yes, that means that model rockets could be designated as explosives. Regulated drones. Projects involving unmanned aircraft systems, drones, must follow all state, federal, and country laws. Radiation. Projects involving radionuclides, radioisotopes, and X-rays must involve a careful examination of the risk associated with the study, and appropriate safety precautions must be taken. Depending on the level of exposure, radiation released from these sources can be a health hazard. All studies may not exceed the dose limit set by the Nuclear Regulatory Commission of 0.5 milligrams per hour or 100 milligrams of exposure per year. Additional information may be found on page 20 of the 2021 SSEF Rules and Guidelines. Guidance for risk assessment in reference to chemicals. Here are brief definitions of 4 categories of possible hazardous chemicals. Those categories are toxicity, reactivity, flammability, and corrosiveness. We encourage all students to be responsible to the environment in reference to use of chemicals. Please encourage the students to prevent waste, use of safe possible chemicals and their products, design of the least possible hazardous chemical synthesis, use of renewable materials, use catalysts in order to minimize chemical usage, use of solvents and reactions, conditions that are safe, maximization of energy efficiency, minimization of accident potential. Form 3 is required when a student or researcher works with potentially hazardous, dangerous equipment and or devices that require expertise to ensure their safe usage. For example, the use of power tools by students that usually do not use power tools should be supervised and documented on Form 3 and in the research plan. Form 3 is required when a student researcher works with radiation sources that produce radiation beyond what a student would normally be exposed to in everyday life. A common example is the use of an ultraviolet light source. The information presented here and additional information may be found online at websites for the International Science and Engineering Fair and the Florida State Science and Engineering Fair Rules and Guidelines. Thank you.
Human participant studies have always been fraught with all kinds of tricky areas that can present lots of problems for science research students. This year, in the year of COVID, it is particularly that way. So when we're adopting human participant studies to our COVID world, the very first thing we have to concern ourselves is with safety. Obviously, the coronavirus passes from person to person, and when we're dealing with human participant studies, that is typically a person-to-person issue. So at the SRC this year, when we were looking at this issue, we wanted to make sure that our very first priority was safety. We, nor you, want to be the cause of some student getting infected because of doing their science research project. So we have a few new rules this year, and they're in effect for this year only. The very first one is that all human participant studies must be done remotely. That means all virtual, no individual student to research participant contact. And that goes for absolutely everything. And also we want you to be aware of all your local health and school board regulations, because those are also going to be incredibly important this year to make sure that everyone stays safe. Informed consents and parental permissions are extremely important again this year. They have to be collected remotely. That's going to require a lot of effort on your part and your students' part. All signatures on the consent forms have to be electronic or digital. We will allow a scanned consent form that has been signed to be sent as an email. However, and this is also important, we want these to be sent to the qualified scientist or to the designated supervisor, not to the student. Again, in an abundance of caution, we want to make sure that students aren't sending their emails out to anyone that wants to get them or can use them in a nefarious way. Now, we do have one exception for this, and we realize that we're late to the game here. So anybody that has consent forms approved and collected prior to October 1st will still be able to use those consent forms. Now, that doesn't mean data collection or anything else can be done other than electronically, but those consent forms that have been approved prior to October 1 will be good. And the next thing we want to be concerned about is just talking about, in general, recurring areas of concern with human participant projects. This is year in and year out the largest area for projects that fail to qualify. And the three basic issues, one is making sure there's a prior IRB approval. The IRB has to be approved prior to this consent forms going out and prior to any data being collected. The next largest problem we have is in the proper makeup of the IRB. The IRB is to be as local as possible. So if your school can put together an IRB, that is going to be the one that is most cognizant of the concerns of your community. And therefore, that's the one that ISEF wants you to use. Now, if you're unable to make up your own IRB, then your regional director can help you in putting together an IRB. And the other one that we come into problem areas oftentimes is the use of humans when the student is doing an engineering product or a product testing and they fail to get consent. If your student is put together something and is going to have humans participate, test it, again, they need prior IRB approval. And the bottom line on all of this is the rules are in the ISEF rules. And there's also an ISEF risk assessment. On this next slide here, you see the two links to those sites. Please use them. Please review them with your students to make sure that they are complying. And please, above all, stay safe.
Hello, my name is William Furiosi, and on behalf of myself, Dr. Nancy Webster, and the rest of the State Scientific Review Committee for the state of Florida, I'm here to talk with you about potentially hazardous biological agents, or PHBAs. Potentially hazardous biological agents are any studies involving bacteria, fungi, or viruses. In any of these studies, it is your responsibility to determine the risk assessment and biosafety level. A biosafety level defines particularly the level of harm, injury, and disease to plants, animals, humans, and involves where you identify the assessment of the biological agent itself, but also the actual procedure involved in culturing and performing experimentation with that biological agent. It also will determine the level of containment needed, meaning where you would work with it, either in a BSL-1 or BSL-2 level laboratory, or the qualified expertise of the adults who need to supervise and or be responsible for the oversight of the project. A little bit more into biosafety levels. Biosafety levels, again, involve both procedures and the microorganisms, the PHBAs themselves. BSL-1 studies, including the use of E. coli K12, must be conducted in a BSL-1 certified laboratory. Within that laboratory, you must make sure that you have all proper protective equipment, including lab coat, goggles, and gloves, and it cannot involve any procedures where you might open up a petri dish or subculture. Once the initial culture is made, it must remain sealed until it is disposed of properly. BSL-2 studies cannot be performed by any junior division researchers. They must also be conducted in a certified BSL-2 laboratory. Just like any PHBA project, you must wear appropriate protective equipment, again, including lab coat, gloves, and goggles. You must also be supervised by a qualified scientist who possesses at least a master's degree or higher in microbiology or pure science related to that field with specific training in microbiological techniques. In this instance, somebody who has a degree in education, master's or above, is not going to be qualified to be a qualified scientist for a BSL-2 project. BSL-2 studies also include any procedure that involves the opening of a culture vessel after inoculation. So if you take a BSL-1 study, you then reopen that container, that makes it a BSL-2 study. In the state of Florida and throughout the entire International Science Fair circuit, BSL-3 and BSL-4 studies are prohibited. Regarding PHBA minimum required expertise, BSL-1 projects must be supervised by a designated supervisor. That supervisor must have at least a bachelor's degree and training in microbiological techniques, including culturing and aseptic technique. BSL-2 projects must be supervised by a qualified scientist. That qualified scientist must, again, have a master's degree or higher in microbiology or a field related to microbiology, and also have specific training in those techniques. Again, individuals with a master's level or higher degrees in teaching or education is not appropriate for these studies and cannot serve as a qualified scientist. For all studies involving PHBAs, PHBA projects must have prior approval by the Scientific Review Committee in order to make sure that they properly ascertained the BSL level regarding the organism and the species, and that their procedures are appropriate for those levels. It also must be conducted in a laboratory environment, not at home. This is especially important when it comes to most students being at home due to COVID. That means that they will unable to do a PHBA project unless they have access to a laboratory. Anybody working with PHBAs must have personal protective equipment, again, including lab coat, gloves, and goggles. They must also be familiar with aseptic technique, making sure to have their lab environment sterilized before and during and after conducting their experimentation, while also properly conducting culturing techniques as necessary and inoculation techniques. Students must have a research plan which documents those aseptic techniques in detail. They must also document proper disposal techniques in detail. Information according proper disposal techniques can be found within the SSEF rulebook within the PHBA section. It must also have standard microbiological procedures referenced in the bibliography, indicating that the student and the designated supervisor, sponsor, qualified scientists are all aware of how to properly handle PHBAs within the laboratory. The International Science Fair rules allows states and other organizations to adopt stricter standards than what they put forth. The state of Florida does so regarding PHBAs. Prohibited research in the state of Florida includes any research involving coronaviruses. At the junior division, it is prohibited to work on any viruses. Students are prohibited from using wild collected mushrooms. Multi-drug resistant microbes are also similar to, as we see here listed, MRSA, VRE, and the others, and other related resistant microbes. They are prohibited from research by students in the state of Florida. Additionally, projects designed to produce multi-agent or multiple drug-resistant strains are prohibited. In this case, it is commonly used selectively to have an individual drug resistant, but if you were to add additional drug resistant genes into that organism, that would be a prohibited project. It is prohibited to subculture from microbial fuel cells unless conducted at a regulated research institution. And projects involving emerging pathogens carried by arthropod vectors like mosquitoes and flies are also prohibited. Some special notes regarding PHBAs. Naturally occurring plant pathogens may be studied at home, but may not be cultured or introduced to a home or garden environment for fear of its spread. A project must include PHBA procedures and paperwork if microbes are used in any way in the project, even if the focus is on something else other than the PHBA itself. So for example, if a student is working with C. elegans, because C. elegans requires a culture of E. coli to feed upon, that means that you must grow E. coli in order to feed and grow your C. elegans. So please make sure that you include PHBA considerations there. Additionally, unknown organisms collected from the environment. An unknown organism is considered BSL1 if it is cultured in a plastic petri dish. That dish remains sealed throughout the entire experiment and is disposed of properly. It is BSL2 if that petri dish is opened or further subcultured. Regarding paperwork, it's important to include the following forms. For example, Form 2, a qualified scientist form, is required for all BSL2 projects, and it is also just a good thing to have to make sure that somebody is overlooking your PHBA research. It is important also to have a risk assessment Form 3, a Form 6A, potentially hazardous biological risk agent form, a BSL1 or BSL Level 2 checklist based on the procedures and or nature of the organism you're working with, and if research is being conducted in a regulated research institution or industrial setting, it requires a Form 1C. Here's what a Form 6A looks like. This form 6A must be completed by the designated supervisor or qualified scientist before any work is started. This is important to make sure that before anybody uses a PHBA in their studies, that they're conducting it safely and appropriately. This also must be included in the paperwork for review by your local SRC. For students conducting PHBA projects at non-school sites, the student is performing PHBA research at a location other than the student's school. Your project paperwork must include the following. Before you begin research, you must include that BSL 1 or BSL Level 2 checklist. And new due to COVID, a letter of pre-approval on institutional letterhead by the appropriate administrator, dean or department chair for a college or university, must be provided with complete contact information. That same administrator must also sign the BSL checklist. An important note as well is the person who signs off on this letterhead of approval in the BSL checklist cannot also serve as the adult sponsor, the qualified scientist, the designated supervisor, or on the school's SRC for the project. After completion of the research, because this is done at a non-school site, a Form 1C would be needed. And again, as a reminder, no PHBA projects can be conducted at home at any level. So if you have any questions regarding PHBA procedures, please ask us, and definitely do so before the research begins. We are more than happy to help screen procedures and make suggestions. I hope you found this video helpful, and again, please reach out to us if you have any questions.
This section of the presentation is going to be covering projects that involve recombinant DNA studies. The first two slides on this talk about some safety concerns for all potentially hazardous biological agents, and they were actually covered earlier in the presentation. We wanted to include those two slides just as a review for you, but we're not going to actually talk to you about them. Please make sure you look over them though before you start your projects, because they are important, especially this year with all of our concerns with COVID. So our presentation discussion's actually going to start on slide 3. When we talk about safety and whether or not students can do an rDNA study, the very first thing we look at is what is the organism that a student will be studying. If the organism that a student will be studying is classified as a BSL-1 organism, and it's going to stay a BSL-1 organism throughout the project, then that entire project can be conducted in a BSL-1 lab under the supervision of a qualified scientist or a designated supervisor. It does still have to have prior SRC approval because it's a PHBA project, but it can be done in a BSL-1 lab following all of the normal BSL-1 safety procedures. Now, if a student is wanting to do a project that starts out with a BSL-1 organism that during that procedure converts into a BSL-2 organism, then that entire procedure, the entire project from beginning to end has to be done in a BSL-2 lab. You can't switch over to a BSL-2 lab after that organism makes the change to BSL-2 status. From the very beginning of the project to the very end of the project, the entire thing must be done in a BSL-2 setting under the supervision of a qualified scientist this time, because this is now a BSL-2 project. It still requires SRC prior approval, and now the safety requirements are more stringent as well. So if the organism starts as a BSL-1, converts to a BSL-2, the entire project is now considered a BSL-2 project. If the initial organism is a BSL-2 organism, then this entire project has to be done at a regulated research institution. This project is going to have to be done under the guidance of a qualified scientist, and it's also going to have prior approval from the regulated research institution itself. They have a governing body called an IBC. They'll review the project and decide whether or not the student can do that project at the institution. This year that's a little bit tricky because of COVID, so allow some extra time if you are thinking of doing this type of project. There are some projects that we don't want students doing at all. Any type of project working with our DNA that involves toxins are prohibited for student research. Doesn't matter if that toxin is a human toxin, plant, or animal toxin, we do not want students doing that, even under the supervision of a qualified scientist at an RRI. So toxin projects are completely prohibited. There are genome editing studies that can be done in a BSL-2 setting. For example, CRISPR can be done in a BSL-2 setting, but there are other genome editing studies that have to be done at a regulated research institution, and we've listed some of those on the second bullet. Please make sure that you are looking carefully at what it is the student wants to do, and that you are certain of which of these techniques they're wanting to use. If they're wanting to use one that has to be done at a regulated research institution, then once again, there has to be a qualified scientist involved. It'll require prior approval from the institution's IBC before that student can begin working. We've always stressed that any plant or animal material that has been experimented on cannot be released back into the environment. And we know that students and teachers and directors understand that, but anytime we're talking about genetically altered material, there needs to be extra care taken to make sure that accidental release does not happen, because certainly we don't want anything that's been genetically altered to even accidentally make its way out into our environment. So extra care needs to be taken when we're looking at genetically altering any type of living material. Thanks very much.
Here are the state's guidelines for working with vertebrate animals, and more specifically, non-human vertebrates. Students are encouraged to use alternatives to vertebrate animal research. If vertebrates are included in student research, the rationale must be discussed in the research plan. Guiding principles for the use of animals in research include replace vertebrates with alternative experimental groups, reduce the number of vertebrate test subjects without compromising statistical validity, refine the experimental procedure to eliminate pain or distress to the animals, and respect animals and their contribution to research. Vertebrate animals are live, non-human vertebrate mammalian embryos or fetuses, tadpoles, bird and reptile eggs starting 3 days or 72 hours prior to hatching, and all other non-human vertebrates, including fish, at hatching or birth. Zebrafish embryos are the exception and may be used up to 7 days or 168 hours post-fertilization. Required of all vertebrate studies would be a research plan which includes justification for using vertebrates, the reason for choosing the particular species, the source and number of test subjects, a discussion of considered alternative test subjects and why they were not used, an explanation of the contribution the research will make to the field of biology, a detailed description of methods and procedures including experimental design and data analysis, a discussion of plans to reduce discomfort, distress, pain, and injury, and the identification of species, strain, sex, age, weight, source, and number of animals proposed for use. Approval required of all vertebrate studies includes having prior approval by a scientific review committee. The approving SRC must include a veterinarian or animal care provider with training in the species being studied. Projects carried out at regulated research institutions must be reviewed and pre-approved by the institution's Institutional Animal Care and Use Committee. Vertebrate animal research must satisfy U.S. federal law as well as local, state, and country laws and regulations of the jurisdiction in which the research is performed. Prohibited research includes research projects which cause more than momentary or slight pain or distress. Any condition or illness of weight loss must be referred to a veterinarian for medical care. This must be recorded by the designated supervisor, qualified scientist, or veterinarian. If caused by the study, the research must be terminated. No deaths are permitted in any group or subgroup. The State Science and Engineering Fair requires that a mortality form be presented with the paperwork for every vertebrate animal project. Projects that are designed or anticipated to cause vertebrate death are prohibited. Any death must be investigated by a veterinarian or other qualified professional to determine if the cause of death was due to the experimental procedures. If a death is the result of the experimental procedure, the study must be terminated, and the study will not qualify for competition. Weight loss or growth retardation of more than 15% is prohibited. Other prohibited vertebrate studies include toxicity studies, behavioral experiments using conditioning with aversive stimuli, mother and infant separation or induced helplessness, studies of pain, and predatory prey experiments. Cautions. Justification is required for an experimental design that involves food or fluid restriction. If the restriction exceeds 18 hours, the project must be reviewed and approved by an institutional animal care and use committee and conducted at a regulated research institution. Animals may not be captured from or released into the wild without approval of authorized wildlife or other regulatory officials. Fish may be collected from the wild only if the researcher releases the fish unharmed, has the appropriate licenses, and adheres to state and local and national fishing laws and regulations. Electrofishing is permissible only if conducted by a trained supervisor. For the purpose of husbandry responsibilities, experimentation beginning date is established as the moment you take possession of the subject animal. Any action taken involving obtaining and setting up the experiment is included in the experimental responsibilities for the vertebrate. And the example would be setting up aquaria. Supervision. A qualified scientist or designated supervisor must directly supervise all research involving vertebrates except for observational studies. The SRC approval process must be repeated if the researcher makes any procedural changes after initial SRC approval before experimentation can resume. Vertebrate research sites can be conducted at home, school, agricultural properties, in the field, and at zoological collection sites. These must have SRC prior approval with a veterinarian or animal care expert in membership. They must adhere to both of the following guidelines. A. The researcher involves only agricultural, behavioral, observational, or supplemental nutritional studies. And B. The research involves only non-invasive and non-intrusive procedures. Vertebrate projects conducted at regulated research institutions have additional requirements. All research conducted at a regulated research institution must have an institutional animal care and use committee review and approval before experimentation begins. These projects must be carried out under the supervision of a principal investigator who is affiliated with the regulated research institution. In addition, student researchers may not perform euthanasia. Required forms for vertebrate projects include the State Science and Engineering Fair entry form, the State Science and Engineering Fair abstract, Form 1, the checklist for adult sponsor, Form 1A, the student checklist, the research plan and project summary, which includes a bibliography that has an animal care reference. Form 1B is the approval form. Form 1C is the regulated research institution form if it applies. Form 2 is the qualified scientist. Form 3 is a risk assessment. Form 5A is a vertebrate animal form for home, field, and school research. Form 5B is the vertebrate animal form for a regulated research institution research. And then the State Science and Engineering Fair Mortality Report. Ask us. If you have any questions regarding vertebrate procedures or projects, please ask us, and do so before the research begins. We're more than happy to screen procedures and make suggestions. And we really hope that these guidelines will come in handy for you. Thank you.
Hi, my name's Maya McGuire, and I am going to be talking to you about environmental projects that may have some concerns or issues. One group of these projects is water collection for testing. And our main concern here is that students may be exposed to the toxins that are released by several types of harmful algal bloom forming species. I need to mention that it is not always obvious if there are toxins in the water. And there are times when it looks like there probably should be toxins in the water because the water is pea soup green, when there may not actually be toxins in the water. So bottom line, you cannot tell by looking at a water sample whether it may have these harmful toxins in place or not. So we err on the side of caution. And we have decided that the younger students, the juniors, may not collect their own water samples. They may test those water samples, but the actual collection must be done by an adult supervisor. Older students, seniors, may collect and test their own water samples unless there is an obvious algal bloom. That would be shown by discoloration in the water. All students, whether they are juniors or seniors, must discard their water samples within 24 hours after collection. And sometimes we're asked, well, how should I dispose of my water sample? And there is no one size fits all answer because the answer depends on, well, what have you added to that water? If it's simply water that was collected and hasn't had any chemicals or anything else added to it, then it can just be disposed of down a sink or down a drain. But if chemicals have been added, you need to research the chemicals, their SDS, and their disposal procedures, because some of those may not go down the drain. It's important to have an understanding, and those SDS sheets should be referenced in the bibliography for the project. We are aware that it is possible to purchase different algae, including cyanobacteria or blue-green algae. Again, our main concern is with the potential for bloom-forming species that can release toxins to potentially be in the samples. Therefore, again, we have decided that juniors may not culture or experiment with cyanobacteria, period. Whether they're wild collected or purchased beyond that 24-hour time period. So we don't want there to be an opportunity for the algae to potentially proliferate and produce toxins. If seniors want to work with blue-greens, they need to do so in a registered research institution if the experimentation is going to take longer than 24 hours. And it's important that they know what genus and species they are working with and that they're using the appropriate protective equipment. All of that should be laid out in their research plan. Switching gears, we get a number of projects with invasive plants. And in the state guidelines, it references using the FLEPC list. This is the Florida Exotic Pest Plant Council's list of invasive species to determine whether a permit might be required to work with certain species. Unfortunately, the most recent list, the 2019 list, FLEPC has removed the category, the column that tells you whether certain plants are restricted or not. So you need to have students use the 2017 FLEPC list. In the Gov list column, it will have either an F or the letter U to distinguish between the plants that have a Florida listing, which is designated with the letter F, or if they are federally listed as noxious weeds or other restricted species, in which case they have the letter U. If there is a letter in that Gov list column, the student should check with the Florida Department of Ag and Consumer Services to see if they will need a permit to work with those plants. They also should ask about the disposal methods for those plants, because again, there is no one size fits all. Disposal method is going to depend on the type of plant that's being used. Invasive animals are a little trickier because even if students collect invasive animals in the wild in Florida, things like Cuban tree frogs or green iguanas, those animals may not be released when their experiment is finished, and nor may they be euthanized. So this gets tricky because they are vertebrates, and how can students work with them and then not release them, not dispose of, or not euthanize them? So students working with invasive animals should contact FWC ahead of time to come up with a game plan for turning over these animals to FWC when they're done with the project. Collections involving marine or freshwater animals need to follow FWC, our Florida Fish and Wildlife Conservation Commission, regulations. So some students will be familiar with the fact that they do not need a fishing license if they are under 16. However, they do still need to follow guidelines as far as size and numbers of species that can be collected. They also need to be aware we get a number of shellfish projects, especially related to water quality, using oysters. Students need to be aware that there are areas that are open for shellfish harvest and areas that are closed for shellfish harvest. And legally, they cannot collect clams or oysters in areas that are closed for shellfish harvest. They may be able to get approval from FWC, in which case they should have a letter accompanying their paperwork that grants them that approval. But with a regular fishing license or following regular fishing rules, shellfish cannot be collected from closed areas. So there's a website that they need to go to check to see if where they want to collect is open or closed. Another thing that students should keep in mind is when they are doing field collections of plants or animals, they need to fill out the risk assessment form, and they need to be aware of all of the potential issues and risks that are associated with doing field collections, and they should address those. And a really important thing, if they are collecting on any property that is not their own house, yard, private property, they need to document permission from landowners or managers. This is true for state lands, for things like parks, county properties, as well as other private properties. And again, that documentation needs to be in with their paperwork.